So welcome to another session. So we have started discussing with a few genetic disorders. So in the previous session, we started off with genetic disorders, right? So in today's session, let us continue with the same genetic disorders, right? So in the if you could remember the previous session, we saw that all the genetic disorders could be classified into two major categories, right? One is called the gene disorders and the other one is called as the chromosomal disorders. Gene disorders, we also call it as Mendelian disorders, or we also call it as inherited disorders. So these are the disorders which are actually inherited from one generation to the other generation. And those sort of disorders are actually usually seen by birth, right? So this is what we have with gene disorders. So we started off discussing some sort of gene disorders. So we in turn saw that the gene disorders could be in turn classified into two categories. One is called the autosomal gene disorder and the other one is called the X-linked gene disorder or sex-linked gene disorder. So under autosomal gene disorders, we in turn again made two different classifications. One is called dominant autosomal gene disorder. Second one is called recessive autosomal gene disorder. So in continuation with this, in the previous session, we took up with autosomal dominant gene disorder. Right? So those were the things what we took up. So a couple of diseases also we have seen in detail, right? Diseases such as myotonic dystrophy, Alzheimer's disease, Huntington's disease, or Huntington's chorea. So these are the three disorders what we have taken up with regards to autosomal dominant disorder. And we have started off with autosomal recessive disorders also. So with regards to that, we have took up two disorders in the previous session. Both are said to be metabolic disorders, right? So one was actually called as alkaptonuria. The second one was phenylketonuria. So these are the two autosomal recessive disorder what we have seen. So in this session, let us take up with a couple of autosomal recessive disorders more, and we'll start getting into some of the details of these autosomal recessive disorder. Right. So the third autosomal recessive disorder, what we'll be taking up is, right. So we have with autosomal recessive disorders. So in the last session, we have taken up with two autosomal recessive disorders. One was alkaptonuria. I told you that. That was the first identified genetic disorder, alkaptonuria, by a scientist by name Richard Garrod, right? Archibald Garrod, right? So we have also seen what exactly is this alkaptonuria, how it has been caused also, and also what is the reason for alkaptonuria also. Similarly, we saw about this also, the other disease also, which is called phenylketonuria, which is also considered a metabolic disorder, right? So in this session, let us take up the third disorder in the list, right? So the third disorder in the list, which is called as cystic fibrosis. It is called as cystic fibrosis, right? Simply referred to as CF, right? So the next disorder under the category autosomal recessive disorder, what we're going to take up is this cystic fibrosis, simply abbreviated as CF. So cystic fibrosis is also considered as an autosomal recessive disorder. So what does that mean? There is some sort of a recessive mutation, right? So there is some sort of a recessive mutation seen in the genes located on the autosomes. So here, the mutation is actually present in a gene that is present on chromosome number 7, right? Gene located on chromosome number 7, right? So on gene that is located on chromosome number 7, there is this mutation. So this mutation leads to this sort of a condition which is called as cystic fibrosis. Now, what exactly are the characteristic features of cystic fibrosis, or what happens in this condition called as cystic fibrosis? So what exactly is happening? As I've said, there is a gene that is located on chromosome number 7, and due to that mutation in that particular gene, there is a condition called cystic fibrosis is actually seen. Now, what actually happens is, due to the mutation in the gene that is located on chromosome number 7, right, a defective glycoprotein is actually formed, right? A defective glycoprotein is formed. So what does this mean? Defective glycoprotein, improper glycoprotein, right? The protein which is not functioning properly is being formed because of the mutation, right? So due to mutation in the gene located on chromosome number 7, a defective glycoprotein is actually being formed. Now, what does this defective glycoprotein do? So this defective glycoprotein will start synthesizing or will start secreting a thick, viscous mucus, right? So this defective glycoprotein will now start synthesizing or secreting a thick viscous mucus. Now, this thick viscous mucus that is secreted, right, will go and get accumulated in some of the important ducts that are present in the body. Right? So what exactly is happening here? Due to the mutation in the gene of chromosome number 7, right, a defective glycoprotein is being formed. So due to this defective glycoprotein, a thick viscous mucus is being secreted, and this thick viscous mucus will get accumulated in important ducts that are present in the body, right? So in the ducts such as the liver, ducts of the liver, ducts of the pancreas, ducts of the respiratory tract such as the lungs, the bronchi, the bronchiole, and to some extent even in the ducts of the skin also. Right? So this thick mucus that is being secreted will go and get accumulated or deposited in the ducts of all of these major organs that are present in the body. So the ducts of the liver, in the ducts of pancreas, in the ducts of lungs, as well as even in the ducts of skin also. As a result of which, what would happen? The entire duct would be completely blocked. Right? As a result of which, there is no movement of blood or the other substances happening because of the blockage of these ducts with this thick mucus that is being present. Right. Now, along with the blockage, especially in case of pancreas, right? Especially in case of pancreas, what happens is there is fibrous cysts that are formed. Right? So fibrous cysts are formed in pancreas because of this viscous mucus, right? So a fibrous cyst is being formed. Since fibrous cysts are formed in the pancreas, we start calling it as cystic fibrosis, right? So this is the condition that is actually associated with cystic fibrosis due to mutation of a gene that is located on chromosome number 7. Right? So what is happening in this cystic fibrosis condition? We said that it is an autosomal recessive disorder. Right? Since it is an autosomal recessive disorder, so what does that mean? There is a mutation in a gene located on the autosome. Here it is chromosome number 7. Right? So due to mutation of a gene that is located on chromosome number 7, a defective or a faulty glycoprotein is being produced. As a result of which, thick viscous mucus is being secreted. Right? So that thick and viscous mucus will get accumulated in the ducts of organs such as the livers, pancreas, organs of the respiratory tract such as the lung, the bronchi, the bronchioles, and also even in the ducts of the skin also. So due to this accumulation, the entire ducts of these organs would be completely blocked. As a result of which, there is no transportation of substances or even blood also happen. Right? Now, in case of pancreas, these thick mucus would lead to formation of fibrous cysts in the pancreas. Therefore, we start calling it as cystic fibrosis. So this is one condition that is associated because of the mutation of this particular gene. There is one more condition also associated with cystic fibrosis, where in this condition what happens is there is improper production or a faulty transmembrane conduction protein is being produced. Transmembrane conduction protein. Right? There is a faulty or a defective transmembrane conduction protein that is being produced. So what does this mean, transmembrane conduction protein? So the proteins that are responsible for conduction of ions across the plasma membrane, we call them as transmembrane proteins. Right? Transmembrane is nothing but something to do with transportation across the membrane. Right? So the protein that is responsible for transportation of ions across the membrane is not produced properly. A faulty protein is being produced. So that protein is referred to as the transmembrane conduction protein. So because a faulty transmembrane conduction protein is being produced, what would happen now? There is no transportation of ions happen. Ions such as the sodium ions and chloride ions are not getting transported properly because of this faulty protein. Now, if they are not transported properly, what would happen? They would be seen excreted out in the sweat. So presence of sodium ions and chloride ions would be seen in the sweat. Right? So this is another condition that is associated with cystic fibrosis. Right? So the second condition, what did we say? Because of this mutation, a defective or a faulty transmembrane conduction protein is being produced, as a result of which the transduction, or rather the conduction of ions such as sodium and chloride ions is not happening properly. So because it is not happening properly, whatever sodium and chloride ions are there in the body would be sent out in the form of sweat. So the sweat of these people suffering from cystic fibrosis will have more concentrations of sodium and chloride ions. So this is what we have with the third disorder under the head autosomal recessive disorder, which is called as cystic fibrosis. Right? A quick recap of what we have seen with cystic fibrosis, then we'll move on into disorder number 4. So what happens in cystic fibrosis is, as I've already said, it is an autosomal recessive disorder which is caused due to mutation in the gene located on chromosome number 7. Right? So because of this mutation, a defective glycoprotein is being formed. And due to this defective glycoprotein, a thick viscous mucus is being secreted. Right? Thick viscous mucus is being secreted. Now this thick viscous and mucus, right, will go and get accumulated in the ducts of organs such as the liver, the pancreas, the lungs, as well as the skin. Right? So once they get accumulated in these ducts, so what would happen? All of these ducts would be completely blocked. Since all these ducts are blocked, right, so there is no proper transportation of substances happening across these particular organs or across those particular ducts. Right? Second condition associated with that is, right, so this thick mucus, right, starts forming fibrous cysts in the pancreas. The third condition is, due to this mutation, a faulty transmembrane conduction protein is being formed. Right? So because a defective transmembrane conduction protein is being formed, the transportation of sodium and chloride ions across the membrane would be affected. Now, since the transportation of sodium and chloride ions are affected, what would happen? More amounts of sodium and chloride ions would be sent out of the body, or it would be rather excreted out of the body in the form of sweat. Right? So this is what we have with the third disorder under the category autosomal recessive disorders, which is called as cystic fibrosis. We have this, right? So let's move on with another disorder, disorder number 4, right? Disorder number 4, which is called as sickle cell anemia. So we have seen about this disease a couple of times, right? So let us again get into some more details with regards to the genetics of this sickle cell anemia, right? So this is the disorder number 4. It is called as sickle cell anemia. Most of the cases it will be written as SCD, which is called as sickle cell disease, or rather sickle cell disorder, right? Either sickle cell anemia or sickle cell disorder, right? So from the name itself, we can make out what is the condition. Sickle cell, right? So the shape of the cell, instead of round, that's what we saw in the previous sessions also, we said that the shape of the RBCs, which is a biconcave disc-like structure, will become sickle-like structure. Right? So that is what we call it as. We refer to as sickle cell. And when we talk about anemia, what is anemia? Lower concentrations of or decreased concentration of hemoglobin in the body is referred to as anemia. Right? So a sickle cell anemia means due to sickle-like formation of RBCs, right? Anemic conditions is seen in that particular individual. Anemic conditions in the sense lower concentrations of hemoglobin in the body. So that is what is referred to as sickle cell anemia. Now, this sickle cell anemia is also an autosomal recessive disorder, right? If you could remember, we have taken it as an example of point mutation, right? So we have taken it as an example of point mutation, right? So what is this point mutation? If there is change in, right, if there is change in only one nitrogen base, we start calling it as point mutation. So that same point mutation is actually seen associated with this sickle cell anemia, right? So this sort of a mutation is seen in the gene that is responsible for synthesis of beta polypeptide chain, right? For the synthesis of beta polypeptide chain of hemoglobin, right? So the mutation or point mutation is actually seen in the gene that is responsible for the synthesis of a beta polypeptide chain of hemoglobin. And this gene is located on chromosome number 11, right? So the gene that is responsible for the synthesis of beta polypeptide chain is located on chromosome number 7. Right? So a point mutation in that particular chain would lead to this condition called as sickle cell disease, or rather sickle cell anemia. Right? So what exactly is happening? So if you could recollect the structure of hemoglobin, right? Hemoglobin falls under the category of a quaternary protein. Right? So if you could start recollecting the topic called as biomolecules, what you have taken up in the first year, we would have studied about the quaternary structure of protein. Right? So one of the examples that we give for quaternary structure of protein is hemoglobin. Right? So when we say hemoglobin is a quaternary structure of protein, right? Hemoglobin has 2 alpha chains and there are 2 beta chains in case of hemoglobin. Right? So hemoglobin has 2 alpha chains and 2 beta chains. These 2 alpha and 2 beta chains are arranged in a heteropolymeric condition. Right? They are arranged in a heteropolymeric condition. Right? So that's how they are actually being arranged. So each hemoglobin molecule consists of 2 alpha chains and 2 beta chains, which are arranged in a heteropolymeric structure, heteropolymeric condition. Right? Now, mutation in one of the beta chains or one of the beta polypeptide chain would start leading to this particular condition called as sickle cell anemia. So what exactly is happening? Right. So if you see the beta polypeptide chain, there are 6 amino acids which are present. 1, 2, 3, 4, 5, 6, 7, and it goes on like that. Right. So there are several amino acids that are being present. So we are actually interested in 6th amino acid. 4, 5, 6, and 7. Right. The 6th amino acid, which is a part of the beta polypeptide chain, usually codes for an amino acid called as glutamic acid. It codes for glutamic acid. This is in normal condition, right? In normal condition, the 6th amino acid, which is present in the beta polypeptide chain, is glutamic acid. But in this disorder called sickle cell anemia, amino acid glutamic acid is being replaced by another amino acid, right? It is being replaced by another amino acid which is called as valine. Right? So glutamic acid is being replaced by another amino acid which is called as valine. Why it is being replaced? Because of this point mutation. So if you just go into details of where exactly the point mutation is happening, right? So if we see in case of normal conditions, right? We'll first take up the normal conditions, then we'll get into the sickle cell condition. So if you see in case of normal condition, in normal conditions, the DNA sequence would be C, T, and C. So we are talking about the DNA sequence here, right? So the DNA sequence is said to be CTC. Now, if DNA sequence is CTC, once this undergoes the process of transcription, right? I think all would have studied about transcription and translation. So once this CTC strand undergoes the process of transcription, right? So the sequence that is seen in case of mRNA would be C will become G here, T will be A here, and again C will become G here, right? So this is the sequence on the mRNA after the process of transcription. So after transcription, the next thing what we have is the process called as translation. So this is what we start calling it as central dogma of molecular biology, right? So we have DNA getting converted to RNA through the process called as transcription. mRNA would be again transformed into proteins through another process called as translation, right? So through the process of translation, this GAG, right, will now start coding for an amino acid which is called as glutamic acid, right? So this is usually seen in normal conditions, where on the DNA the sequence is CTC, on the mRNA it would be GAG, right? So after the process of transcription, the mRNA will now start coding for an amino acid called as glutamic acid. This is in normal conditions. Now, in case of sickle cell anemia, what is happening is, right? What's happening is this particular strand, that is the DNA strand, here there is point mutation happening. As a result of point mutation, what is happening? This T here, thymine here, is replaced by A. Right? Thymine is replaced by adenine. Right? So the sequence here would be the same. It would become CAC. So CTC has been transformed into CAC in case of this diseased condition. Right? Once it has been transformed into CAC, the point mutation is between T here as well as A here. Now, because it has become CAC, the mRNA of this, right? The mRNA of this would become, C will become G here, and A would become U here because it is RNA, and C would again become G here, right? So what is happening now? GAG is being transformed into GUG. Now this GUG codon, once it undergoes the process of translation, it would now start coding for an amino acid called as valine. Right? So this is what is happening. So glutamic acid is being converted into valine in this particular condition. So since the sixth amino acid, which was actually glutamic acid, through point mutation with just one nitrogen base change, what is happening now? Glutamic acid is being completely transformed into valine, or valine starts replacing glutamic acid in the sixth amino acid position of the beta polypeptide chain. Clear this? So this is what is happening here. So let me quickly go through it once again, right? So we know that hemoglobin has 2 alpha chains and 2 beta chains. So these 2 alpha and 2 beta chains are arranged in a heteropolymeric nature. They are arranged in a heteropolymeric condition. Right. So in the beta chain of hemoglobin, 6th amino acid in normal conditions usually codes for glutamic acid. But in this condition, that is sickle cell anemic condition, what's happening is the glutamic acid is being replaced by valine. Right? Glutamic acid is being replaced by valine. How is it being replaced? By observing this, we can identify that in normal condition, the DNA sequence would be CTC. This CTC will undergo the process of transcription to form the mRNA having a sequence of GAG. So C will become G here, T will become A, and C will become G. Now GAG will now start coding for an amino acid called glutamic acid through undergoing a process called as translation. Right. Now there is a point mutation in the DNA sequence, right, where T is being replaced by A. Thymine is being replaced by adenine, whereby the sequence now will become CAC instead of CT and C. Now, since it has become CAC, in the mRNA, it would become GUG. Now, this GUG will now start coding for another amino acid, which is called as valine. So, valine gets replaced by the original glutamic acid. Now, because valine is being replaced by glutamic acid at the 6th position in the beta polypeptide chain, the beta polypeptide will not produce a proper, or beta polypeptide chain is not being produced properly. As a result of which, hemoglobin is not produced properly. There is some sort of distortion in the structure of hemoglobin. Since there is distortion in the structure of hemoglobin, the RBC having such sort of hemoglobin, instead of being a biconcave shaped RBC, will now start turning into a sickle shaped RBC. So this was normal condition, and this is sickle shaped RBC. Right. So this is what is actually seen in case of sickle cell anemia. right? So this is all is happening on the gene which codes for beta polypeptide chain, which is located on chromosome number 11. Right. Now, what would be the effects of sickle cell anemia? What effects are seen in the individuals suffering from this sort of a mutation or this sort of a condition called as sickle cell anemia? So let us take up with the conditions that are seen associated with sickle cell anemia. The first thing, hemoglobin structure is being distorted, right? So that is what we have said, right? Number one, hemoglobin structure is distorted, gets distorted, or it gets altered. We can just take it down as altered, right? So hemoglobin structure gets altered. Now, because the hemoglobin structure gets altered, what would happen to the structure of RBC? So RBC would become sickle shaped. RBC would become sickle shaped, right? So this is the second condition that is seen. In the first condition, what is happening? Hemoglobin structure gets altered because of this point mutation. The second thing that happens is the shape of RBC changes and it would become a sickle shape, right? So once the RBC's shape becomes a sickle shape, what would be the outcome of it, right? The oxygen binding ability to hemoglobin, right? Oxygen binding ability to hemoglobin will decrease. Hemoglobin decreases, right? So the oxygen binding ability to hemoglobin will start decreasing, because we know that during the transportation of oxygen, oxygen usually binds to hemoglobin and forms something called as oxyhemoglobin. Now, since hemoglobin structure is altered and at the same time RBC also it has changed its shape, the oxygen binding ability to hemoglobin, or rather RBC, will start decreasing. Once it starts decreasing, what would happen? The oxygen concentration in the body will start decreasing, right? So once oxygen concentration in the body will start decreasing, what would happen? Our cells and tissues in the body will not have sufficient amount of oxygen for carrying out a process called as cellular respiration. As a result of which, it would lead to the death of cells or a particular tissue, and very soon it would lead to the death of that particular individual also. Right? So this is the outcome of sickle cell anemia. So that's the reason why we say the individuals suffering from sickle cell anemia will not survive until the reproductive age. So before the reproductive age is reached itself, they would die because of low oxygen concentration in the body. Right? So these are the outcomes of this condition called sickle cell anemia, which is considered as a autosomal recessive disorder. Right. Now, getting into the genetics part of sickle cell anemia. Right. So we said that the gene that is responsible for beta polypeptide chain, the mutation in this gene is responsible for sickle cell anemia. So how do we denote that gene? So the gene is actually denoted as HBA gene. So if the condition is HBA and HBA, so these two are dominant alleles. Since it is said to be a dominant allele, this individual will be normal, seen in most of the cases, right? So if this is the condition where the alleles are both dominant alleles, so there is a HBA allele which is dominant and another HBA allele, both are said to be dominant, and that condition, the individual will remain normal. The second condition we have, there is a mutation in one allele. So mutated gene in the allele, we call it as HBS, and this also will be HBS. So if there is HBS, A is a dominant allele, S is a recessive allele. This is how we start denoting a dominant and recessive alleles in case of sickle cell anemia. HBA and HBA are said to be normal. That is HBS and HBS. So this is said to be a recessive condition where the individual will start suffering from sickle cell disease or sickle cell anemia, right? So this individual will usually die before the reproductive age is attained, right? So before reproductive age is attained, this individual usually die. So there is one more possible condition here, which is heterozygous condition. Homozygous dominant, individuals will survive. There is no problem at all. Homozygous recessive individuals will die before the reproductive age because of sickle cell disease. The third condition is the heterozygote condition, where it is HbA and HbS. So there is one dominant allele here and one recessive allele, right? So these individuals, we start calling them as carriers, right? We start calling them as carriers for sickle cell disease, right? So we start calling them as carriers for sickle cell disease because there is both a dominant allele as well as a recessive allele, right? So as I've already said, this individuals, there is no problem because they are normal, they don't have the disease. This individual, both are recessive alleles, so they usually die before the reproductive age. Now, the problem arises for these sort of individuals, who we call them as heterozygote individuals, where there is one dominant allele and one recessive allele being present. Right. So if this is the condition, there are certain things that these individuals has to follow. Right. Try to understand this properly. In normal conditions, when there is enough of oxygen available, right? In normal condition, when there is enough of oxygen available, this individual will start producing normal RBCs. Right? So as long as enough oxygen is available, right? So these individuals, who we call them as carriers, will start producing normal RBCs. But once they move to the place where the oxygen tension is low, right? Try to make a note of this. Once they move to the place where the oxygen tension is low, at that point of time, this mutated allele, that is HbS allele, will now start undergoing a process of polymerization, right? So they start undergoing a process of polymerization and they will start producing sickle-shaped RBCs. They'll start producing sickle-shaped RBCs, right? So what exactly is happening? As long as these individuals are carriers, they are at a place where there is enough of oxygen availability, right? In those places, this individual will start producing normal hemoglobin molecules. Thereby, normal RBCs are also being produced. But if they move to a location or a place where the oxygen tension is low, right? So once they move to the place where oxygen tension is low, what would happen? This mutated allele, HbS, will now start undergoing a process called polymerization. So through this process, they will now start producing sickle-shaped RBC. Right. So if for these individuals, they should always be, if they have to survive for a longer period of time, they should always be present in a place where there is enough of oxygen. So if they move to the place where there is oxygen deficiency or low partial pressure of oxygen is said to be low, at that point of time, this particular individuals, along with normal RBCs, they will start producing even the sickle-shaped RBCs also. So this thing they have to be very careful with where they are actually being present. Right? So that is one condition associated with this. Now, these sort of individuals, that is, we start calling them as carriers. These carriers for sickle cell anemia, they have one advantage also. Right? Even though they are very much prone to this sort of a disorder, that is sickle cell anemia, they have one advantage also. Throughout their lifetime, they will never get the disease called malaria. Right? So throughout their lifetime, they will never get the disease called malaria. So these individuals, that is carriers for sickle cell disease, right, would be resistant to malaria throughout their lifetime. So why they would be resistant to malaria? Because we know that if for malarial parasite to undergo the process of reproduction, it requires RBC. Right. Now here there are chances that sickle-shaped RBC also would be produced, whereby the replication process of the malarial parasite would not happen properly. So these individuals, they are said to be resistant to malaria. So throughout their lifetime, they will not get this disorder called as malaria. So this condition or this effect, we we start calling it as heterozygote advantage. Right? It is called as heterozygote advantage. So what exactly is this heterozygote advantage? In a heterozygous condition, these individuals that are said to be heterozygotes will be or they will start attaining some sort of advantage. And here, with regards to sickle cell anemia, that advantage is they will not get malaria throughout their lifetime. Right? So this is what we have with sickle cell anemia. Right? Nowadays, what scientists have identified is, right? Nowadays, what the scientists have identified is with regards to this particular carrier. With regards to this particular carrier, they have identified that HbA and HbS both are said to be co-dominant alleles. Right? You remember the topic called as co-dominance, which we took up in a previous sessions before, right? Co-dominance. So what happens in co-dominance? Both the alleles will individually start expressing their phenotype. So here also, HbA will start producing normal RBCs, whereas HbS will start producing sickle-shaped RBCs, right? So HbA is producing normal RBCs, HbS will start producing sickle-shaped RBCs. So both are said to be dominant to each other. So nowadays, the scientists, after doing much work on the sickle cell diseases, by identifying the genotypes, they came to a conclusion that this particular carriers for sickle cell disease, that is in a heterozygote condition, they are considered to be co-dominant for each other, co-dominant for each other. Right? Now, along with that, we also put this gene as an example of pleiotropic gene. So pleiotropy also we have discussed a few sessions before. So what did we say in case of pleiotropy? If one gene is responsible for more than one phenotype, then we started calling it as pleiotropic gene, right? And that condition and that sort of inheritance, we call it as pleiotropism. So here it could be taken as an example of pleiotropic gene also. How it could be taken as an example for pleiotropic gene? Number one, same gene, it is responsible for producing normal, biconcave, saucer or disc-shaped RBC. At the same time, the same gene is also responsible for producing sickle-shaped RBCs. So it could be taken as an example of pleiotropic gene also. So two things with regards to the carriers, or rather three things with regards to carriers, what we need to remember. Number one, carriers for sickle cell disease are resistant to malaria. They will not get malaria throughout their lifetime. So this condition, we start calling it as heterozygote advantage. Second thing is, this carriers for sickle cell disease can be also taken as an example of co-dominant, because HbA will start producing normal hemoglobin, HbS also will start producing sickle-shaped hemoglobin. Right? So both individually they'll be start producing their RBC. So this carriers for SCD, that is heterozygote condition, could also be taken as an example for pleiotropic gene. So what is pleiotropic gene? If one single gene is responsible for more than one phenotype, then we start calling it as pleiotropic gene. So the same pleiotropic gene could be seen even here also. Right? So this is what we have with sickle cell anemia, which is considered as an autosomal recessive disorder. So disease number 4 under the list called as autosomal recessive disorder. Right? I hope everything is clear till now so far. Right? So quickly let us take up with one more disorder, right, for today. Right? And that disorder will be disorder number 5. Again, the same thing under autosomal disorders only. Right? So under autosomal disorders only, let us take up another disease which is called as albinism. Right? I hope you have heard about albinism also. Right? So disease number 5 is called as albinism. So what exactly is this albinism all about? So albinism is nothing but lack of pigmentation on the skin. So if you could recollect when we were talking about skin inheritance, skin color inheritance with regards to polygenic inheritance, we said that in polygenic inheritance, if all the 6 alleles are dominant, maximum amount of melanin is being produced. If all the alleles are said to be recessive, what did we say? There is no melanin secretion at all. Therefore, we started calling that individual as albino individual. So the individual of that particular process of getting albino individual is called as albinism. right? So albinism is also a autosomal recessive disorder, which is caused due to mutation in a gene located on chromosome number 11, right? Mutation in a gene located on chromosome number 11. So what happens during the process of pigmentation is, we know that in humans, the skin color is because of the secretion of a pigment which is called as melanin, right? So the skin color in individuals is because of the secretion of melanin. If more amount of melanin is secreted, the individual will be darker. If less amount of melanin is secreted, the individual will be fair. That is what we know. And we have also seen that this melanin secretion is genetically controlled, right? And the type of inheritance that is shown by this is polygenic inheritance, what we have taken up a few sessions before, right? Now here, for the melanin secretion or synthesis, right? So a metabolic process has to occur. So if you could recollect what we saw in the previous session with regards to a disorder called phenylketonuria. So the same thing, continuation of that, we can get to this, right? So what happens is, we have a amino acid which is called as phenylalanine. A-L-A-N-I-N-E, phenylalanine. So this is one of the amino acid what we have already seen even yesterday also, right? So this phenylalanine, under the influence of an enzyme called phenylalanine hydroxylase, it would get transformed into another amino acid called as tyrosine, right? So phenylalanine, in the presence of an enzyme called phenylalanine hydroxylase, would be transformed into another amino acid called tyrosine. This is what we saw in the previous session when we were discussing about phenylketonuria. In phenylketonuria, what did we say? There is a mutation in the gene which is responsible for synthesizing PAH. As a result of which, phenylalanine is not getting converted to tyrosine. That was phenylketonuria. Now we are talking about albinism. So what happens in albinism is, the precursor is the same, phenylalanine. Phenylalanine, in the presence of phenylalanine hydroxylase, would be transformed into tyrosine. First stage. Second, this tyrosine now, in the presence of an enzyme called tyrosinase, right? In the presence of an enzyme called tyrosinase, will start forming a pigment called as melanin, right? So this is how melanin is actually being formed. So this is the normal process of melanin formation, right? Where phenylalanine first gets converted into tyrosine, and tyrosine now gets converted into melanin in the presence of an enzyme called tyrosinase. Now what happens in this condition, that is albinism, is the gene that is responsible for synthesis of this enzyme, tyrosinase, is being mutated. Right? Getting it? Right? So the gene that is responsible for the synthesis of the enzyme called tyrosinase is being mutated. And that gene is located on chromosome number 11. Now, because of mutation in the gene coding for enzyme called tyrosinase, what is happening here? The enzyme tyrosinase is not produced. So if the enzyme tyrosinase is not produced, what would happen? There is no melanin secretion. So if there is no melanin secretion, it would lead to albinism, where there is no pigmentation, right? Because of albinism, or because of no melanin, there is no pigmentation, right? There is no pigmentation seen in skin, right? There is no pigmentation seen on the skin. At the same time, there is no pigmentation in hair. There is no pigmentation in iris of the eye also, right? So there is no pigmentation seen on the skin. There is no pigmentation on the hair, and there is no pigmentation in the iris of the eye also. Now, since there is no pigmentation in the iris of the eye, in majority of the cases, people who are suffering from this disorder called albinism, they have no vision, right? So there is loss of vision because there is no pigment seen in the iris of the eye, right? So this is another disorder. We start calling it as another metabolic disorder, which is called melanism, right? Another metabolic disorder. inborn metabolic disorder, right, which is albinism. So with regards to inborn metabolic disorders, we have taken up 3 disorders. One is called alkatonuria, phenylketonuria, and the third one is albinism. All 3 fall under the category of autosomal recessive disorder, right? So three disorders we have taken up for today, right, which is cystic fibrosis. First one, due to mutation in the gene located on chromosome number 7. Second one is sickle cell anemia due to mutation in the gene located on chromosome number 11. And the third one is albinism, again caused due to mutation in the gene located on chromosome number 11. Right? So we have seen what exactly is happening in each of those conditions, where the mutation is seen, and what are the outcomes of those particular disorders. Right? So in the next session, we'll take up with another disorder what we have under the head called autosomal recessive conditions. Right? Then we'll move on into the X-linked disorders. Right?